Okay, hello everyone. I'd like to welcome you all to the Advanced Therapy Seminar on behalf of the Cell and Gene Therapy Catapult in collaboration with the University of Oxford. So first of all, I'd like to thank everyone for attending. And just to say, I think the aim of this seminar really is to provide kind of some information and kind of education, I guess, around some of the key considerations around taking kind of later stage academic research through to a commercial product, and what considerations need to be taken in place, and what to do with that information. So before we move on to the speakers, I just want to do some general housekeeping. So just to ask everyone to display your Zoom name. Oops, sorry. Check your Zoom display name. Unmute yourself for the Q&A session we'll have slightly later on, and in the breakout rooms, and if obviously you're asking a question, you can either keep your video off or on. That's up to you. Please use the raise your hand function just to kind of keep things orderly. That really helps. As part of this seminar, what we're going to do is have breakout rooms for 3 separate but concurrent discussions. So to allow everyone, and what we're going to do is allow everyone to choose which room they can go into. But to be able to do that, we have to allocate you in there. So what we're going to ask everyone to do is put a number in front of their name, so they will be allocated into that particular room. And to do that, it's quite simple. So you go next to your name, click on More, select Rename, and then we're going to ask you to either add a 1, 2, or 3 in front of your name to designate which room you'd like to go into. And then one of my colleagues will put you into that room. So hopefully quite straightforward, but let us know if you're having any difficulties. So this is the agenda for this afternoon. Introduction by myself. We're going to have then a talk from the Catapult CSO. That will be followed by a case study from Professor Robert McLaren. We'll then have a series of topic introductions by our breakout speakers. They're going to be about 5 minutes each, and they'll allow you to select which room you want to go into. We'll have a short kind of comfort break, and then put everyone into their breakout rooms. And then after the breakout room, we'll have a panel discussion. So yes, so next thing to do is to hand over to Jonathan.
Okay, thank you very much. Just bear with me a second while I share my screen. Okay, Sam, can you confirm you can see that, please? Thank you. Good afternoon, everybody. Welcome to the seminar. Thanks very much for joining today. So I'm going to give just a short, approximately 15-minute presentation about the background of the Catapult, with a particular focus on how we want to increase the amount of support we give to the commercialization of research to improve translation of new technology through into UK industry. So many people will have heard of the MRC or BBSRC, the funding agencies which allocate grant money to support UK R&D, but not so many people have heard of Innovate UK. And Innovate UK are a parallel funding agency, but slightly more focused on the growth of industry in the UK. And Innovate UK manage the network of catapults in the UK, whose role is to support the growth of industry. And as you can see on the right-hand side, there's about 10 catapults working in a diverse range of areas which are innovative of their nature. And of course, the Cell and Gene Therapy Catapult is the one here presenting today. As I mentioned, we're part of a network, and our role is to develop facilities and expertise that we can really apply to help the translation of research, but also the growth of companies that are in this space. Today, as I've mentioned, it's going to be a little more focused on what we do to help with translation, e.g. bridging that gap between academic research and businesses, with a focus to adding value, moving material out. But there is also a role for the Catapult helping industry reach down into academic research for the help that they may need with their own development. But an important point that I want to emphasize very strongly is that the catapults are not-for-profit businesses. And that's important, particularly when we work in this translational environment, because we are not here to try to exploit the technological concepts that you've got. And I think that's an important way for people to view us, because we can give help without trying to take something in return, if you like. It's not like a venture capitalist type support structure where you have to give something in order to get something. We are simply here to try to do what we can to help you grow, help companies grow, and help translation industry. So within the cell and gene therapy environment in particular, there are kind of three main areas of challenge, probably taking them in relatively reverse order of the amount of resources that the cell and gene therapy catapult allocates to each of them. From a health economics perspective, if any of you have been to any of the seminars focused on cell and gene therapy, there's a lot of discussion about reimbursement models where you give a treatment once and it adds value, gives value to the patient for many years. How do you really capture that in terms of the health service, which just doesn't have the kind of systems in place really to allow that long-term demonstration of value to work? But also we also try to help people starting new projects to understand where that could fit in the market and what kind of competition that they might expect, and the costs of development, to really work out the economics of whether they have a viable business opportunity. ATMPs have their own bespoke regulatory and clinical framework. ATMP legislation, as it's called in Europe, or CGT legislation in the US. And that's largely as a recognition of the fact that the manufacturing challenges are quite bespoke, and the risks are somewhat different with antibodies or more traditional platforms. So we have about 30 members of staff focused in preclinical and clinical regulatory areas to help companies navigate that space, or academics to plan about how they might navigate that space. But the majority of our resources, as you'll see on the next slide, are really focused on addressing the manufacturing and supply chain of cell and gene therapy. So many different platforms, vectors, cells, autologous cells, different types of vectors, in vivo, ex vivo, and the way to manufacture these drug substances is bespoke to every project that we find. So what have we developed in order to enable us to support the industry? On the left-hand side of the slide here is a caption describing our development centre laboratories in London, which is the area that I focus on. We've got 1,200 meters squared purpose-built centre, and we focus on what you would expect, the development of novel analytical techniques, helping companies or academics develop manufacturing processes which are robust, which can be scaled to higher productivity levels to support later stage or commercial development. We do a lot of work on viral vector manufacturing, not so much the discovery of vectors for therapeutic purposes, but really how to manufacture third parties' discoveries. We work in stem cells, and we also have recently opened a collaboration area on the 10th floor, so we can work side by side and give access to the equipment that we have. We're growing famous for the manufacturing centre in Stevenage, which is a 7,700 square metre facility where third-party companies can come into the manufacturing environment that the Catapult manages, and they can bring their bespoke manufacturing processes, their staff into that environment, have their own clean room, when we will collaborate with them to help them develop any processes that they might need, and we can share the costs of managing that centre across the different collaborators we've got. And we've got companies like Freeline, Autolus, TCR Squared, and Adaptimmune in there, all making different materials, some making lenti, some making AAV, some making autologous cell therapies, all under one roof. And you may have seen recently that we became involved with what we've named the Manufacturing Innovation Centre. This is a bespoke facility to support the COVID-19 international effort. It's unusual for us to be in this space. It's principally there to support vaccine manufacturing as part of the national effort, but it's possible that we could also support some cell and gene therapy in there after the vaccine effort is done. So, Sam, I can't see a clock, so I don't know if you can tell me how much longer I've got left.
You've 9 minutes.
Okay. So this slide very much recapitulates what's on the previous slide, but I just wanted to emphasize something that's not covered on the other slide because it's particularly relevant for the discussion today. We've got these physical capabilities which look very good in pictures, but we also have a large group, 30 or so, I think it's 35 actually, members of staff under Jacqueline Barry, some of whom you'll meet today, that give regulatory, non-clinical, clinical support to the Catapult staff when we're developing our own programs with third parties, but also to third parties, companies that need help navigating this area. So now I just want to finish the presentation on a single slide which talks about our growing, changing strategy to support commercialization of research in the UK. What we're finding and increasingly recognizing is that there's a pretty decent funding system in the UK to help academics that have got good differentiated ideas, to test them in vitro models to demonstrate that the mechanism of action is there, maybe develop some biomarkers, maybe test them in vivo models. But as the costs of the in vivo models get more increase, particularly for cell and gene therapies, this is a kind of cut-off point at which it becomes more difficult to begin to translate these concepts to the next stage. And what we believe is that in the UK, investors are looking for a lot more than that. They want to see that this idea can be supported by a reliable, scalable manufacturing process. They may want to see preclinical safety pharmacology data generated to prove that it will translate into the clinic. They always want to see an experienced and committed team behind that vision, and they want that team to understand how their product is going to fit within the medical environment, what kind of costs it will take to deliver the plan through to clinical proof of concept. And they're always concerned about freedom to operate on the platform. And this area, if you think of this like a traffic light with rather more light bulbs than you would normally see, is the way we're beginning to approach projects. And we think that the Catapult can help, particularly with these ones towards the bottom, clear vision of the medical product, realistic plans to deliver proof of concept, health economic med affairs modeling, some background freedom to operate, rigorous freedom to operate, as Matthew will explain, is a big undertaking, but we think we can provide some support in this area. And then there's those top two, a reliable, high-yielding manufacturing process and preclinical safety farm. We can help advise on those. We can actually do them, but then it gets really expensive. We're often talking in the realm of £1 million for each of those two yellow bullet points there. And realistically, we might help 1 or 2 companies a year in that space if we're lucky, but most of the time it's going to require grant or VC funding. So really what we're looking to do is to find people like yourselves with these great differentiated ideas, some convincing in vitro data, potentially with in vivo models, measuring translatable endpoints, and then help you fill in the rest of those traffic lights so that when we go to VC companies together, you've got a really good cohesive story that will help you attract the investment that you need to take your product through to a company and successful. So that's it from me. I'll stop there, and I believe either I'll hand over directly to Rob to tell his story, or Sam, perhaps you'll take care of the next step, please.
Thanks, Mary. Thanks very much, Jonathan. Well, I'll first just ask if there's any questions from the audience. As I say, please raise your hand if there are. Otherwise, we'll jump straight into Robert's presentation then. So Robert, thank you. Take it away.
Good. Thank you very much, Jonathan, for a very clear summary and introduction. It's great to be talking about the Nightstar gene therapy program again. It's quite nice to be doing it sitting down at home as well, which is quite unusual. But anyway, I'd just like to really talk a little bit about the work that we've been doing as academics, and how that sort of became a big commercial program. And I guess the sort of things I've learned on the way about how academics can contribute to this research, and the sort of things that I think that we can do very well to help the companies. So we're working with adeno-associated virus, which is a non-replicating parvovirus, and it contains within it a single strand of DNA. There's no cell membrane around adenovirus, so it's generally very low immunogenicity. And it is particularly effective at transducing neurons, or as the case that we're interested in transducing retinal cells, cells in the retina at the back of the eye. We have in the genome the replication and capsid gene. This is the wild type virus, and we can remove those genes, which gives us approximately 4.7 kilobase of cargo capacity that we can then put in the virus, and the virus will then deliver that gene safely into neuronal cells, retinal cells. And this disease that we're particularly interested in at the beginning, choroideremia, is caused by a deficiency of a protein known as REP1, and the coding sequence for REP1 is 1.9 kilobases. So the retinal degeneration that you see on the left, the atrophy, leads to blindness, and it's X-linked, so it causes blindness in young men. It's progressive. They initially have normal vision in childhood, and then gradually the visual fields constrict. And you can see the degeneration on the picture of the retina on the left compared to the normal retina on the right. So the goal was always to put the missing gene into the adeno-associated virus and deliver it to the retina of these unfortunate people in order to try and slow down or stop the progression of the disease. So when designing viral vectors, you need to think about a number of factors to make the gene express efficiently. And again, this is work that we do in the lab a lot. And I think that we have a lot of experience in the basic science of vector biology, and we can troubleshoot many of the problems ahead of the commercialization process. For instance, you can see here on the left, you can see, let me just move that. On the left, you can see the promoter, the beta-actin promoter, and then we've got EIE, which is the intron-exon splice site, and we put that in deliberately to enhance expression of the gene. We've got a ribosome binding site and Kozak consensus, which assists with translation. Here's the REP1 sequence with a start codon and a stop codon. And then we've additionally added this thing, WPRE, which is another viral element, the woodchuck hepatitis virus post-transcriptional regulatory element, which assists again in enhancing the gene expression. And one of the things we did was we made the gene expression as efficient as possible, because if your viral gene expression is very high, you will need less dose of virus to get the same treatment effect. And the less virus that you inject into someone, the less likely you are to have any side effects. And a lot of this optimization we did at the beginning in the lab in Oxford. This makes messenger RNA, which you can see, which would then undergo translation in the cytoplasm of the cell. And the RNA expression, as far as we know, in non-dividing neuronal cells is likely to be extremely long-lasting, possibly even lifelong in non-dividing cells. It's very exciting gene therapy, a concept of actually one treatment potentially to stop a disease with lifelong effects. Now, when we went to the regulatory authorities, we had to provide data to show that the vector that we were using, that it worked. And we have actually here some example slides from my lab. On the left, you've got the Western blot in B, and on the right in C, you've got immunohistochemistry in the mouse retina showing expression of the REP1 protein in green. And what you see on the left is an experiment using cells taken from humans with ethics approval, of course. The Rep1 expression, which you can see in these bands here on the Western blot. And just below, this biotinylated Rab6A is a functional test of the Rep1 protein to show that it's functional. So what we have here, if you think about it, is we've got two models showing the vector efficacy, both the human in the cell line and also in the mouse model, showing appropriate expression in the retina, which you can see in the target area. And this is really sufficient for regulatory approval. A lot of people told me at the beginning that, oh no, you'll need to do stuff in a non-human primate and all this and that. And at the end of the day, it's really quite simple. If you can show that it works and it expresses the protein as you expect, the only true disease that you can test it in is the human disease. And a lot of the animal models become irrelevant. So that's a good example. We did this work in our lab to GLP standards, all our equipment validated, calibrated regularly, and as a result, the quality of the work in the lab was sufficient to be accepted by the regulators for approval to start the phase 1 trial. We also did the surgery, developed the surgery slightly to inject the virus under the retina, and you can see here just a diagram showing that the virus is injected at the back of the eye, underneath the retina. And the results of the first trial were very successful. The first few patients in the UK in Oxford showing improvements in their vision. And as a result, the program was subsequently spun out to form Nightstar. Nightstar then set up several other trials. And again, these trials became global, involving several sites across the US and in Europe, and all based on the initial work that we did in Oxford. And most of these trial sites were with collaborators and colleagues with whom we've worked as academics over many years. And again, it's very difficult sometimes for companies to create this sort of network, but we as academics intrinsically have these networks in the people we work with and collaborate with on a regular basis. And again, this was very helpful for us when we actually came to commercialize the research to find other trial sites to work with us on the program. And you can see now, even in South America, we have patients coming up for surgery on the East Coast and West Coast of the US. So the program is now in phase 3. It's all very exciting, and it's currently being run by Biogen. Now, the next trial that we worked on is X-linked retinitis pigmentosa. This, you can see, is the photo of the back of the eye, of the retina. And you can see on the left there with the black areas showing the areas of degeneration. And on the right, you can see a cross-section through the retina, known as an OCT scan. And this, again, shows degeneration and thinning of the retina peripherally. This is due to mutation in a gene known as RPGR, which is another X-linked gene. And this is probably the most common of the severe forms of retinitis pigmentosa with huge global impact. Retinitis pigmentosa is the most common cause now of untreatable blindness in young people. And so there's a huge unmet need. And this particular gene is probably the most prevalent of the severe forms. So this is the coding sequence for the DNA, the cDNA for the RPGR gene. And I noticed that there's a few genetics boffins listening in. I can't see them, but I've seen a few names there. And I'm sure they will instantly recognize the problem with this gene. They do talk about complicated slides in PowerPoint, and I have to say I'm quite pleased with this one. I don't think I could get it any more complicated. But if you look down at the bottom here, by the way, you can see at the top there, the start codon, ATG, and at the bottom, there's a stop codon. And if you look in the middle here, that area there is comprised almost entirely of A's and G's. So this gene has evolved with this very unusual sequence in it. And when you have DNA, which normally has 4 genetic code, when you've just got repeated sequences just of two nucleotides, it's very prone to mutations. And these mutations are very common in disease. And also, of course, it makes it extremely difficult to make a viral vector because cloning that sequence is virtually impossible. So the work we did in the lab in Oxford was to change this codon sequence to stabilize the A's and G's and introduce a few C nucleotides to get it in a position where it could be easily cloned into a stable form for gene replacement. And the other difficulty about RPGR is that the splicing is different in different species and indeed in humans in different cells. And so we also had to overcome the problem that the cDNA of the gene, which would go into the viral vector, would undoubtedly be incorrectly spliced if we got the sequence wrong and put the sequence back again into the human cells. So again, we used codon optimization at looking at the different splice sites to stabilize the splicing as well as the stability of the gene. All this work was done in my lab. We then patented the work as well, and eventually that was published. And because we had optimized and patented the codon optimization, when we came to commercialize this particular program, it was obviously a very attractive one for the gene therapy company Nightstar, because they could see that we'd protected the IP as well as generating the novelty to it. And I think, again, this is one of the great things that we can do as academics, is to look at these very detailed questions. So this is a work from, this is Cristina Martinez, who's working in my lab on this project, showing expression of the RPGR at the correct level, the correct density on the Western blot, and also GT335, which is a functional test of prenylation, which shows that the protein is correctly post-translationally modified. So this led to the first gene therapy treatment of X-linked RP, first in the world, in fact, in Oxford on March 2017. And again, the surgery that we used there was something similar to what we developed for the choroideremia program. And this shows an operation with the injection of the virus under the retina, which I hope you can see there, a little needle. And remarkably, we saw in some of these patients an improvement in visual function. This shows the visual fields. And the visual fields you can see at the baseline on the top left there, showing the red areas where the patients can just about see something. And if you look by 1 month, the red area is increasing quite a lot on the right eye, which you see here on the left. And then by 3 months and 6 months. And I think if you look at that 6-month picture and compare it to the baseline, you know, there's no doubt really that these patients have had an improvement in their visual field. And indeed, the sensitivity measured by this visual field testing was quite significantly changed. We published that work in Nature Medicine early this year as the first example of reversal of visual field loss in retinitis pigmentosa, which was hugely exciting. So, and again, this shows the anatomy, and we looked at, again, looking at the scans, we identified in these patients a thickening of the retina, which we think was due to regeneration of outer segments. As academic ophthalmologists, of course, we're very used to looking at scans and looking at diseases. And again, we're able to contribute additional data to validate the observations based on our clinical skills, which really has helped this program. And finally, we're looking at ways of improving the surgical delivery of the virus. And this leads us to another commercial collaboration with another company, Precise, a Dutch company that works on robotics. And we thought it would be safer and perhaps more predictable to use a robot to deliver the virus into the subretinal space. And this led us to do the first robotic eye operation with Precise, using a system that we'd helped them to develop in order to inject a needle underneath the retina at the back of the eye. And this shows a picture, a video of the robotic system in place, now delivering the gene therapy via a robot. And I never really thought that work in molecular biology would lead me to collaborate with a company that is a robotic engineering company. But indeed it does, because logically you have to think about the disease. What are the next challenges? How can you improve the treatment? And the robotic system is the next stage. Here, by the way, at the back of the eye, you can see the needle. This is actually injecting a solution that dissolves this blood clot. And you can see the needle here. And then on the scans on the right, you can see the retina lifting up very, very slowly. It's a very, very slow infusion. And if you want to deliver the virus into the subretinal space, you need to do so without damaging the retina. And having such control with a very slow infusion is a very, very good way of doing that. And this is currently what we're working on at the moment, using the robotic system to deliver the gene therapy treatment. And there you can see very nicely detached. So finally, I'd just like to acknowledge the huge work of my research team. We have clinicians working on the clinical program, also a lot of very talented basic scientists working on the molecular biology, the viral vector development, all the tests that we need to do to develop treatments that are going to be translatable in clinical trials. Currently, we've pretty much emptied the cupboard of AAV gene replacement therapy, and we're now working entirely on CRISPR gene editing programs, using the AAV platform to deliver these into the retina and treat many more retinal diseases. So that's a very, very quick run through. So thank you very much indeed for allowing me to talk. And of course, I'll be very happy to take any questions in the next couple of minutes.
Thanks very much, Robert. That was a really good presentation. I think a nice example of how academic research can lead kind of directly to, you know, a therapeutic, a therapeutic product that can change people's lives. And I thought it particularly interesting how you talked about the idea of, you know, collaborative networks and how important they are in actually establishing it and taking, you know, these types of projects forward, and they're crucial. So I just see we got 5 minutes, so just see if anyone's got any questions. But I think in the meantime, Jonathan, did you have a question? Are you going to answer?
Yeah, sorry, I don't know how to raise my hand on the web-based app. Rob, I wonder if you could comment a little bit about how did you get the funding to do this work in the earlier stages, and did you get slowed down by that, or was it simple and easy?
Well, getting funding for anything that's innovative is unfortunately very difficult due to the peer review system, which is very much geared against new concepts and very much stuck in rewarding people who have been successful in the past doing pretty much the same old thing. I'm not saying there's anything bad about that. That's just the way it is. So one of the issues I had at the beginning was I needed to get funding, but I knew that some of the referees would say that it would never happen, and that, you know, I didn't have enough data, etc., etc. So I went around the back door really, and I was able to get ethics approval for the gene therapy trial from what was then GTAC, the Gene Therapy Advisory Committee. And then when I went to apply for funding, I had the clinical trial approval, you know, like as a, you know, like a gambit sort of thing. And I think many of reviewers would have loved to have said that it would never get approved based on a mouse model and some cells in the lab. And they were probably desperate to say that, but they couldn't because the clinical trial approval had already been made. And I think, you know, one thing I learned is that a lot of people review grants and they kind of feel that they need to be the regulator. They feel, well, I need to stop this going forward because it could be risky. And a lot of academic colleagues don't understand the difference between a phase 1 trial and a phase 3 trial. And they think that we really need to have enough data to show unequivocal that it's going to work before we start a trial in humans. And again, part of my remit, I guess, is to try and sort of educate others and try and bring everyone on board and just say, look, you know, we'll never really learn unless we do a phase 1 trial. And that's the point where all the excitement really begins, because we've seen amazing things in phase 1 trials that we would never have predicted and would never been able to measure in an animal model. How can a furry mouse tell you that that the mouse is sitting further away from the television set because his visual field has improved so much? You know, that doesn't happen.
Thank you.
Yeah, that's really interesting. I think when I was listening to your talk, I was quite interested in a bit when you said you engage with regulators, because we have that question a lot. At what time, when should I engage with regulators? How early should I do that? And it seemed like that you did that quite early, I think, to obviously get around this issue you had with the grant funding.
Yes, I mean, you know, we need to know from an early stage, and I think, you know, we shouldn't be afraid of the regulators. They are actually there to help us, and in most cases, they give extremely helpful information. And so if you are going down the route to a clinical trial, you can arrange a meeting, you know, obviously it's now done online, where you can talk to a medical advisor or a scientific advisor, talking about the design of your trial, which medical advisor and maybe the scientific advisor would give you advice on the amount of preclinical data that you need in order to start your trial and what the MHRA will be looking for. And this is a very, very well established route. And I would definitely advise anyone who's considering a phase 1 trial to have a pre-meeting, you know, with the regulators before they make the formal submission. It'll save them a huge amount of time in the long run. And that's irrelevant to the funding. That's just very important for the trial setup.
Thanks. We've got a question here from Omar Habib, and it's what were the challenges in patenting your vector?
Well, the challenges, if you think about it, are about prior disclosure. One of the things I've noticed is that whenever you write a paper about something exciting, you know, in the discussion at the end, you say, well, you know, in future, you know, this might be applicable to retinal gene therapy for treating an eye disease or something. And so, you know, in a way, there's lots of papers published in science where there may be an idea in the discussion that it might have a future application. And that comes up quite a bit in terms of proving that your observation really is novel. You need to look at what it is that you've done that's novel. The other thing, I guess, is, you know, I've got a big research group with PhD students, postdocs. You know, it's difficult sometimes. I have to tell them, look, you know, we're going to have to sit on your data for another 18 months, maybe 2 years, until all the patenting is done, and then you can publish your paper. And that takes a bit of, you know, a bit of mentoring. It's quite difficult sometimes for very, very keen, you know, motivated students to just allow their data to sit there whilst the patenting process goes through. But it's crucially important, and I use many examples of where that can happen. And I kind of give them a carrot, and I say, look, you know, you're going to get your name on a patent and you're going to get a paper. So you basically get 2 for 1. You just need to wait a bit longer for it. But again, be careful what you publish, be careful what you present at meetings. We make sure also in our lab meetings that when we're presenting sensitive data, any visitors to the meeting are asked to stand outside whilst it's presented, and everything is kept quite secret until the patent is filed. And then, of course, we can talk about it freely.
Thank you, Robert. That's some really kind of good, helpful words, I think, especially around the patenting. I know Matthew Garner, our head of IP, will no doubt endorse those comments, but we can pick up some of those streams in the roundtable later. So I'd like to move on now to our kind of quick fire presentations from our 3 breakout room hosts. So we'll go for in order, and I think this is important because this is for the number you'll have to put in front of your name. So we'll start with Mustafa, we'll then go with Lee, and then finally Matt. So I'll pass over to Mustafa.
Sorry, I just had to unmute myself and take myself off of video. Please let me know if I run over the 5 minutes, Sam. Hi, everyone. So my name is Mustafa. I'm one of the lead scientists here at the Catapult as part of the industrialization team. A lot of the work we do is around manufacturing. So I'll be chairing the manufacturing breakout room. And so I just want to give you a glimpse of what we're going to sort of be discussing within that. So manufacturing is relevant I assume to most of you, because to test your product, whether that's in vitro, in vivo, or within the clinic, you need to first be able to make it. So we'll be discussing a bit about the different considerations, manufacturing considerations at the different stages of development. So if you've just sort of shown that your concept works and you're looking to sort of get investment, what are the key considerations you might need to be thinking about? If you just started preclinical tox studies, so you're about to go into a phase 1 clinical trial, is anything different there? Are there different requirements? And there are, but also what are the different requirements once you're already in the clinical trial, say your phase 1, and then you're trying to move into phase 2 or your pivotal phase 3 trial. And similarly, if you're about to start your phase 3 trial, at what point do you need to absolutely lock down your manufacturing process? I'm going to also have a bit of a discussion with those in the room, and it's going to be a bit of a tailored discussion focused around what you're developing. So we can cover different ATMPs, whether that's a gene therapy, an autologous cell therapy, or an iPSC derived or an allogeneic cell therapy, because they all have different considerations. So to make most use of the time, the discussion will really be focused around those in the room and what they're actively pursuing and doing. We'll also have a discussion around when to think about scaling. Scaling is going to be relevant. You're not going to need you're going to need a lot more product when you go into a mouse, and then when you go into a human, and then when you go into more humans, etc. So everyone needs to think about scaling, but it's just having a discussion around when. That's really important, because one of the key things is that what you don't want to do is have decisions that you make very early on, really locking yourself into an unviable process. And the trouble with that is if you do have those issues, then what can happen is that you need to go back and do a bridging study, a very expensive in vivo bridging study potentially, and that can make it very unattractive for investors. We'll talk a bit about robustness of your manufacturing process. The further down you go, the more robust the process needs to be, but really want to have a drill down what we mean by robustness and how you can assess the robustness of your process. And again, working with those in the room, we can have a discussion around what you're currently doing, what you're currently using, and maybe what technologies might be more suitable or compatible with as you try to progress to clinical and commercial manufacturing. So do join me, and we'll be able to discuss in a lot more detail all these different areas and anything else that may be relevant that any of you might want to bring. All right, thank you very much.
Thanks, Mustafa. Lee, do you want to go next?
Yeah, just clicking the share screen button. So oops. Let me run that. Hopefully everyone can hear me okay. So in my session, I'm going to talk about the non-clinical side of cell and gene therapy development. And I guess what I mean by non-clinical is the in vitro and in vivo work that you will do during clinical development, but also prior to clinical development. I think we had a previous talk there about engaging early with regulators and getting lots of advice and things. And this talk has a kind of, I guess, a theme running through it, and it's kind of my philosophical mantra, I suppose, which I call biology, biology, biology. we need to understand our therapeutic, because there are no standard approaches to the way you capture data, the way you show efficacy, the way you show pharmacology, the way you show safety for ATMPs. They're all very, very different. And the approach we take is very different from traditional classes of therapeutic products. So I'm going to talk about some of those challenges of developing those programs to get the data you require to initiate clinical development in the first instance. It's a very risk-based approach, so I'm going to talk about understanding clinical risks and then mitigating those clinical risks within a non-clinical package. And I'll illustrate it. So I'll talk about some of the philosophy around it to begin with, but that can sometimes be a little dry. So I've picked a number of examples of licensed ATMPs, and I can talk you through then some of the non-clinical programs that were run that supported marketing of those products. So I'm going to talk about a couple of ex vivo gene therapies and a cell product. The purpose of that, I think, is to really illustrate that there's more than one approach that you can take to generate the data that you need to initiate clinical development. And it's kind of multifactorial kind of approach that it requires very holistic thinking. But at the center of it all is a really good understanding of the biology of the disease, an understanding of the mechanism of action of your therapeutic, and then you build your risk mitigation profile and your package around that. So that's my short introduction. So yeah, please join me if that is of interest to you.
Thanks, Lee. Matt?
Hello, everyone. I will get my screen sharing sorted. Hopefully everyone can see that slide now. So for my quickfire introduction, I'm going to be talking about intellectual property, and in particular the intellectual property considerations that are applicable to the ATMP sphere. And just wanted to reassure everybody that this isn't going to be kind of an intricate dive into the convoluted world of IP law itself and the mechanisms of protection down at the kind of the nitty-gritty legal level, but more an opportunity for us to kind of talk at a fairly high level about some of the IP concerns that arise during the translation of academic research, and ways that we can develop strategies to mitigate risks and to avoid falling into pitfalls. A little bit like Mustafa mentioned earlier, you don't want to lock yourself into certain technical choices at an early stage in development if that's not necessary. Similarly, we can avoid some problems by taking a proactive stance on IP development. So there'll be two main strands that I'll be discussing. First of all, protecting and being recognized for your own work, and also how to deal with IP rights that are owned by others. I was delighted to hear Professor McLaren's comments earlier in relation to the importance of considering strategies around perhaps delaying publication in order to arrange for patent applications to be filed first, and to be cautious about the way in which we frame publications, if this could, certain statements that you make might have an effect on your IP position later on. And of course, you're not going to be the only party that is looking to protect IP rights. There will also be other academic groups, other commercial entities which are working in the area, and so it's almost inevitable that there will be intellectual property rights owned by third parties that need to be considered during the development of a commercial strategy. So within the first part of that discussion, a few topics to be covered will be what types of protection are available, how we might go about recognizing when an invention has been made, and tips for dealing with your tech transfer office or your patent attorney, as may be. And within the second part, a little bit of an introduction to the concepts of IP landscaping and freedom to operate analysis, and some thought about strategies such as licensing of rights using off-the-shelf technology and other options that might be available to address third-party IP rights. I think as for the other speakers, I'd like to kind of present some information towards the beginning, but I would like it to be a reasonably sort of open discussion. And so depending on the specific queries that arise, I can tailor the responses and the discussion to address people's particular interests. And I think that sums up my intentions, Sam.
Thanks, Matt.
Shall I stop sharing my screen now?
Please, yes, yeah.
There we go.
Thank you. Okay, so just to reiterate or just to go over again how you would go about selecting which talk you'll want to be in. Just to say that once you've selected your talk, you'll stay in that particular breakout room. But what we will do, because you may want to obviously be able to understand what's going on in all three of those talks. So what we will do is send everyone a link to a YouTube video of this whole webinar kind of at the end of the day or tomorrow. So you can go back and look what's happening in discussions of the other breakout rooms, so you don't kind of miss out on any of the information that may be valuable to you in those. So what we're going to ask you to do is put a number in front of your name. 1 for the manufacturing talk by Mustafa, 2 for the non-clinical from Lee, and then finally 3 for Matt and his IP talk. So to do that, you go to More next to your name in the participants list, choose rename, and then add either 1, 2, or 3, obviously followed by your name. And then what we'll do is myself and my colleagues will then allocate you to the particular room that you've chosen. If you have any issues, just drop us a line on the chat and we'll try and sort that out. So while everyone's going around doing that, we'll take a 5-minute break just to allow everyone to change their name and sort things out there and have a quick comfort break. And we can pick this up and we'll start the breakout room sessions, let's say about 2 minutes past 2. That gives us 5 minutes. So if everyone can do that, that'd be much appreciated. Thank you. So I hope everyone found that informative and kind of got something out of those individual breakout rooms. Hopefully there was some conversation there as well as a few that I went into. I saw some conversations going around, so that was positive. What we're going to do now then is take maybe some of the lines of discussion and what was raised in those breakout rooms and move into a round table discussion. And so kind of that will be for our panelists, but obviously I welcome all the kind of discussion and questions from the audience as well. You know, we really want to try and address any of the issues you're having specifically and try and tailor some responses to that. But so let's move to that round table now. So the title of the round table is how to build a robust commercialization strategy. So if we can kind of contextualize our answers within that. So I think I'll start off by asking the first question. And I think one of the things that people come to us, academics come to us and ask, well, you know, when do I start engaging with the Catapult? When do I start engaging with CDMOs, investors, whoever, you know, when do I start to think about building that commercial strategy? So I'll put that to the panel. When do you think is the best time an academic group should start to build that commercialization strategy?
Well, I can start from my own experience, Sam. I mean, at the end of the day, I'm a clinician seeing patients. So I'm in a very good position, I think, to design a clinical trial. And if you are working in cell and gene therapy and you are a scientist and you don't have access to patients, then obviously you need to talk to a clinician. And I think it's good to start trials within the academic setting It's very, very motivational for your team. It's a huge asset to the NHS to be involved in clinical trials, wonderful asset for the patients. And I think, as I said to you, we can do phase 1 clinical trials very, very effectively. Even if you don't show a treatment effect, the fact that you've de-risked the trial and the fact that you've got regulatory approval to go into phase 1 adds a huge amount of value. So when the commercial people come in, they'll obviously be much more committed both financially and with their resources to make it go further and hopefully take it through to phase 3 and become an approved product.
I think maybe add a little something quickly to that, Sam. I think there's important considerations that inform your clinical study development that reflect the reimbursability of particular therapeutics. And I think ATMPs are probably quite different than traditional therapeutics, where you would stack them up against standard care. And I think having an understanding of that, having an understanding of how reimbursable it is, having an understanding of the kinds of clinical data that you need to generate in order to satisfy the people making the decisions, people like NICE, and other insurers, is really important. And I think probably with traditional therapeutics, we think of that as quite a late stage consideration. With ATMPs, we start to think of it pretty early in the development process, which then informs a lot of what we do. It informs the clinical protocol, it will inform the non-clinical, and inform manufacturing scale and things, which is pretty useful to understand from the outset.
Can I, Sam, sort of add to that? So, because your question was, so when do you need to start thinking about the commercial strategy and commercialization? So that's after you've done your phase 1, 2, 3, and then you've got your final marketed product, which is, can seem quite far away. And I think it is very far away generally, but I think a lot of what you're doing early on is linked to what you're going to need to do later on. And so I think it comes back to that. Think early on so that you're not blocking yourself later down the line. And we had this discussion in the breakout room that what can you do fairly early on from a manufacturing viewpoint to not block yourself. And then it comes to potentially holding on to retention samples. But one great point that was made is, well, sometimes you can't actually characterize your product because they're so advanced, so novel, and you just don't have the analytics. So I think also thinking about what sort of analytics that you might need to sort of wrap around that is really important, and maybe that's what you need to do early on is actually really be able to really understand your product, because the earlier you do that, the easier the later stages become, and you don't, as Rob was saying, the academics will be really great at doing that phase 1. Perhaps don't need to worry about the phase 2 and 3. You just need to worry whether you've blocked people from easily transitioning to that phase 2 and 3.
I think to wrap up from the Catapult side, I would add that as soon as you start thinking about potential translation of your research into the commercial sphere or into the therapeutic sphere, it's really important to have a pause and give a lot of effort into how are we going to manage the dissemination of this information in such a way that we can maintain the IP position and also achieve the goals of disseminating the information. One thing that I think sometimes is overlooked is that patent applications or patenting as a process is in fact a specialized form of publication. The process requires the publication of the patent application 18 months after it has been filed, and so that can also help to reassure people who feel that maybe patenting as a process is inhibiting dissemination of information. Actually, the opposite is true. It ensures it reaches the public domain. But we need to make sure that the way that the information is disclosed is conducive for maintaining patent protection and for not undermining your own position further on down the line by, for example, disparaging or suggesting that the work that you're doing is in any way obvious or naturally follows on. You always need to make sure that you're when you're filing your applications, you're in a position to say this is fantastic, this is unexpected, this provides great advantages that were not predictable.
Yeah, I think something I thought was interesting you said, Mustafa, is around when commercialization starts. And I think you're right. It probably, if you looked at the kind of dictionary definition, it's probably a lot later, in the process. But I think from an academic point of view, which we've all been there, and many of people in this room are still kind of core academics, that's anything further on from publishing. Basically anything above publishing is, you know, once you get into the realm of patents and thinking about non-clinical studies and manufacturing, it is commercial. So I think maybe it's a where you phrase the definition. But I'm interested to maybe get some thoughts from people from OSI, potentially. I think there's a couple of people from OSI, Angela or Bob, around, you know, when do you think is the best time to engage or to think about your commercial strategy, if we think about that early commercial strategy? And I guess that leads into, you know, when should some of the academics at the university start engaging with OSI and others?
Well, I'll happily take that question, Sam. I'm not sure whether my video is showing there. Just to clarify, I represent OUI, which is Oxford University Innovation, not OSI. That's a separate organisation.
Sorry.
But I think that we tend to take a look at the commercial opportunity and the possible strategy for reaching the market with a potential product quite early. You know, when we're looking at the possibility of filing patent protection on a particular product or technology, we need to look not just at the attributes of that and whether it's inherently patentable and whether there's no prior art that kind of undermines it, but also need to reassure ourselves that there is a market opportunity worth pursuing. So on that basis, we tend to advise researchers at the university to contact OUI quite early and introduce their research so that we can understand what the direction they're going in, even if it's very much a work in progress at that point. And, you know, also advise them on the necessity not to disclose any of their initial research findings prematurely in such a way that it might compromise patenting a little bit later, but also, you know, not signal unduly early to the competition, if you like, which areas they're pursuing. So I think knowing what in this case, the target diseases and indications might be is important from the outset. And often, and this is certainly the case with Robert McLaren, you know, they're leaders in their own areas of medicine and are very familiar with exactly what the markets might be. So it's useful to get that input from the PIs in the project as well. I hope that didn't ramble around the question too much, Sam.
No, I think what you're saying there, Bob, is early is good. So you can provide that support that's needed if people are kind of lacking in that kind of experience with starting to move into that sector. And I mean, another question that I've been thinking about while we've been having these discussions is slightly related to something that came up earlier around funding. You know, a lot of work is funded via grant funding, this early kind of biological research. But you then start to move into the area of having to get funding from private sources such as VC. So I wondered, Robert, maybe you could comment, and I know Lee, you have some thoughts around this as well, around when do you think is the best time to engage with the venture community or maybe angels or other private sources of funding? Yeah.
I think you know, if you're able to get your treatment into phase 1 trial, as I mentioned before, I think this is the ideal time to begin to engage, because the VPs, you know, the venture capitalists, you know, they will see maybe 100 preclinical programs for every one clinical program. So you're immediately going to get their attention. And I think, you know, if you were to, you could spend a long time trying to get funding at the preclinical stage. But as I said, with the clinical stuff, it's actually not too difficult to work out how to do a clinical trial safely, particularly when you're dealing with many incurable diseases that have huge negative impact on human health. So we can get them over that hurdle and then engage. And the other thing I would say is that the reputation of the funder is also an important factor. And one of the discussions I had very early on with Nightstar, having successfully funded the spin out with Syncona Partners, was about who else was going to sort of jump on the bandwagon. And actually, we were quite selective in identifying venture capitalists and others who were a very high repute and very well known in the industry as being people who generally pick winners. And I think that helped us a lot as well, because if you seem to have good backers behind you as well as a good program, that really does help a lot.
Yeah. Lee, did you want to make any comments around that?
I don't think I can add anything that's too different, really. I think the main point is ATMPs are a bit different in the way that they traditionally attract funding. So other therapeutics, venture capitalists can look at the markets and the opportunity and the IP position very readily. I think people struggle a little bit more to get that proof of concept data with ATMPs, and that means it sits in academia for a bit longer, and you're trying to do things with grants and little bits of angel funding to do, to get that early initial clinical study, and that I absolutely agree wholeheartedly with that. That makes it more fundable because you've gone through it. I don't think that's true in the US. I think in the US they are funding at a preclinical stage. I think you're talking about a European kind of UK environment to a certain extent, where they just seem to make things harder for people to get that initial data to attract the kind of big funding. But I mean, I agree with that sentiment entirely. Clinical data just makes you look a lot more credible and investable.
Yeah, that's right. And I think one of the experiences I've had, and I think we've had at the Catapult, is having the conversations with investors is often very valuable, even if you are quite early, because they know the type of questions and the type of issues that are going to be raised later down the line. And so they can actually steer you in the right direction. Even if it's we're not interested for now, it's often a we're not interested now, you should go away and look at X, Y, Z. And so that gives you a really good steer on where you should come back, or what you look at, and then when you should come back to them, and it builds that relationship as well. So I think that is quite valuable. Obviously, if you do have the clinical data, that really validates your approach, and I think you're a lot more likely to get investment. One of the things that I find as well is that they're often looking for intellectual property to be secured as well in the form of patents. I don't know, Matt, what your experience is there.
Yeah, I think it's a generally well-recognized point that if you have registered rights, patent rights, early on in the development of your product or your approach, then you will be regarded as a more serious and more credible opportunity. I mean, it's hard to give sort of hard and fast rules about that, because sometimes it may be that you can obtain the rights that you need through in-licensing. Sometimes it might be that the area that you're working in is more or less amenable to filing new patent applications, depending on the indication that you're looking at. I mean, sort of following on from that comment, one thing I mean there is to say that ATMPs are a bit different from maybe small molecule traditional pharmaceuticals because of the complexity of the product. And so you're not looking typically at a patent for a new chemical formula, which is a new therapeutic molecule. It is, if it's a gene therapy or a cell therapy, it is orders of magnitude more complex than that. And so we don't really talk about necessarily sort of patenting the CAR T cell or patenting the AAV particle. You might be looking at patents which protect specific molecules within those products. So the antibody recognition domain that you've introduced into your CAR T, or perhaps an optimization of the capsid structure for your AAV, or as Professor McLaren mentioned, looking at optimizing the sequence of the cargo that you are encoding into your particle and finding ways of saying, well, this is new, this is inventive, and this is deserving of a patent application. So the patents that we look for in the ATMP space are more like a kind of a mosaic of rights which protect different aspects of the product, rather than saying I have a new patent for aspirin, or a specific molecule.
Thanks, Matt. Another topic I wanted to move onto, and this is something that was raised when I was in the talk that Mustafa was giving around manufacturing, and this is around a particular issue that I know many people have, particularly for manufacturing viral vectors, and that is simply the kind of lack of access to manufacturing facilities. I think this was raised in your talk, Mustafa, and if you want to kind of elaborate on the conversations you had there and maybe some of the solutions that are coming up.
So I think it's one thing that was quite clear is again, the academics can sort of lead clinical trials, and especially phase 1, and then move forward. But what becomes really tricky is being able to get access to affordable manufacturing of these products. And I think that's a problem throughout the field, globally, across the world, actually. But a particularly acute challenge within the UK, and that's something that we at the Catapult have been working with a consortium actually of UK academics and industry, and sort of petition some of the funders, folks like MRC and others, and what we're trying to set up, and what the MRC and LifeArc are now trying to set up, are these translational hubs, in particular for viral vector manufacture, to be able to provide that. So the aim of these hubs will be that they can manufacture GMP vector and be able to manufacture at an affordable cost for academics to be able to access, in particular for phase 1 trials. So it won't be sort of commercial scale, but it will be for clinical trial scale, and so that can be something that the UK academic community in particular can tap into, and be able to request grant funding to be able to support that sort of manufacturing. So hopefully that will be coming through in the next few years, but obviously doesn't help anyone right now. But what we also do is work very closely with a lot of the CMOs and CDMOs within the UK. So I know that Sam, Charlie, and us, we should be able to link you with potentially relevant CMOs or CDMOs who may be able to manufacture your product for you as well for now.
Yeah, that's right. I believe that Oxford are actually one of the put in an application for that particular MRC LifeArc grant to establish one of the viral vector hubs. So fingers crossed that arrives. I think it's going to be 3 or 4 of those hubs around the UK, which I think will be great because it means they're academic led. So it will be led by people who understand the limitations and some of the issues around getting kind of your academic process to a more GMP-like or GMP process. So I think it would be really good. But also having 3 or 4 facilities around the UK will mean that bottleneck that currently exists and the high price that kind of currently exists, potentially having to go to a CDMO or even a CDMO in the US will start to kind of get a bit softer and come down in price and really accelerate things. So I think that's going to be a real big opportunity in the UK coming up.
And I think one of the critical features is actually the technologies that will be going into these hubs. They're not set in stone right now, but the idea that part of the remit is that the technologies that go in will be scalable. They don't have to be commercial scale at that stage, but it should be in a position where anyone who's using these hubs will then be able to go to a commercial scale manufacturer to be able to be supported in transitioning from that small scale to that large scale, should you reach phase 3 and beyond.
Yeah, I think one of the other questions that came up in your talk as well was around, or one of the other discussion points was around, when do you move over to your industrial type of manufacturing process, and should you do that early?
Yeah.
Because if you do it later, there's this issue around bridging studies, having to have retention of samples, and other issues that maybe if you move sooner to a more industrial scale and standards, you get over those.
Yeah, so I think we discussed this both in the context of gene therapies, but also cell therapies, and you know, we always encourage people to move as early as possible, but I having been in academia developing ATMP, so cell and gene therapy products, and having been in industry, so at GSK developing similar products, fully appreciate, fully understand that you're not gonna have you know, a process that's at the scale that would be required for a clinical trial when you're sort of very early on, because you're just not going to get the funding to do that. And so thinking about the cell therapy field, you know, you have a platform, something like the Prodigy and other platforms, which can do everything all in one and then fully automated, but can be very, very expensive. And if you're only needing to make a small amount of, for example, CAR T or TCR-T cells to do your animal studies, then you're not going to use that. So do you want to invest hundreds of thousands in that technology early on, not knowing that your product's going to work? Probably not. And then as you get into your preclinical toxicology as well, do you want to be moving into that at that point, or do you move into it later on? And I can fully see the reason for wanting to move on to it later on, and it's just to say that, well, to really support that, what you might want to do is have retention samples, samples that you can go back to and say, I know this product is safe and works because I used this product in my preclinical toxicology study, and now I could take the product with my new process and really compare them and do an in-depth characterization and say, actually, this product is the same as this product. So regulator, could you please let me go into the clinical trial with this new process without needing to do another toxicology study or start again from an earlier phase, or do a bridging study?
Yeah, and I think I don't know, Leah, I can bring you in this as well, is and to say that some of the issues, I know you're not kind of a key, you know, you're not regulatory specialist, you're doing the non-clinical side, but you might be able to comment on the fact that sometimes when you make a change in your manufacturing process, that affects kind of the regulation as well. So they go hand in hand is the point I'm trying to make it. And I think you've seen that, Mustafa, haven't you? Is when you know, whenever you make a change to your manufacturing process, that has effect on the regulation, and each one feeds into each other. So it's quite important to have that established as early as you can.
I think if you're looking for a comment, Mustafa made the best point, is that keep lots of samples of what you've made, because your quality attributes, so your QC assays will change extensively over time as you characterize your product better and better and better. So at least if you can go back to those early batches, do the additional analytical methods that you've got, then you can do side-by-side comparisons with your new process and show, yeah, we've got some consistency across this. So I think that's really, really good advice if you've got stability of those samples over time. So, you know, viral vector or cells in liquid nitrogen are usually pretty robust. But there is always, if you make major changes to manufacturing processes, that can, you know, result in the need for additional testing. And I think because the manufacturing process really is as much a key component, you know, of the product, you know, as the active pharmaceutical ingredient itself, we have to have really good controls on this. You know, we're building quality into that process when we're developing that manufacturing process.
Yeah, thank you. We've got a few minutes left now, so I wanted to kind of have a little bit of a discussion around some of the non-clinical data that needs to be generated or can be generated. I think, Robert, when you were giving your talk, you said that, you know, often appropriate animal models aren't available. And that, you know, sometimes using human cell lines or using in vitro studies is sufficient because there simply isn't something available there. So I wondered if we could just have a little discussion around kind of how you develop a non-clinical strategy, whether or not you should use animal models, and how you begin to go around kind of developing that side of it, because that's often the next point on from once you have some very interesting proof of concept data. How do I develop that strategy around safety and toxicology and so on?
Well, that's a very interesting point, Sam. I mean, one message I would give to everyone is that you need to know your literature. If you know what studies have been done before, you will find somewhere a study that maybe even used the same serotype of vector that you're planning to use in a human, in a clinical trial, in another disease. And that adds huge weight to the safety and efficacy of your own trial. Now, you know, if we're talking about adenovirus, for instance, or talking about adeno-associated virus, which is the vector I'm most familiar with, the capsid, the outside part of the vector, is pretty much the same. The serotype AAV2 will be the same. And if you think about it, sometimes the ITRs, the promoters, all the other bits inside the regulator is the same. The only thing that's different is that the DNA, the nucleotides in the middle have just sort of been shuffled around a little bit. And so if someone says, oh, we need to do a full immunology screen on that, well, I say, well, look, you know, the immunological component of this is the capsid. That's already been established. And we know that it's been used at different doses in different trials. We really don't need to do another preclinical safety study. And I'm afraid most of these comments come a lot on the commercial sector from someone in a company who's the head of regulatory affairs, you know, someone whose job it is unfortunately to design all these non-stop preclinical experiments that I think in many ways trip up a lot of the early stage companies because they listen too much to what they're told and do that. But in reality, you know, the regulators are common sense people who understand the issues. You have to show your vector is safe as far as you can, and you have to show that it's efficacious as far as you can. Nothing can be proven 100% because we simply don't have the animal models that are the same as the human disease. And of course, the human immune system is quite different to the animal systems. And on top of that, you know, the dose, in fact, there's huge variability, in fact, between individual patients receiving advanced therapeutics as much as there are between species. So if you know your literature, and if you can articulate all these points and show basic safety and efficacy, then I think that's all you need. And I think you'd be surprised at how you may find yourself pushing at a door that is opening for you.
Lee, you raise your hand.
Yeah, I mean, if you read my talk, that's kind of a lot of what I said there, really. Engage with the regulators, they're there to help you. I am one of those regulatory type non-clinical safety people that do due diligence on things and point out gaps that might be a potential clinical risk. However, I think if I can summarize my talk into like just 2 little points, you have to work out how useful any species that you do your non-clinical testing is predictive of human. Yeah, so don't do anything if there isn't a model available. There's loads of literature out there and you can justify a lot of this. Late last week, early this week, it's all blurring into one now, in the MHRA meeting, we went in with a strategy of there is no animal model to do any safety or efficacy testing of this product in. We've got a lot of in vitro data, we've got a lot of clinical experience with this particular type of product in another indication, and David Jones, although it's a confidential conversation, said, I agree, but I'm not telling you what the product is. They are pragmatic and they understand it if you justify it. There's another type of regulatory type person that Robert's talking about, and they sit in big pharma companies and they are totally risk adverse because they get kicked 100 foot up in the air if someone takes in an asset that falls down later on. They don't, that's a metaphorical kicking in the air. And so it's a very risk adverse group of people. And you have to get down to the science, the disease biology, and you know, the understanding of the biology of your therapeutic, and have appropriate conversations around that. Never talk about regulations as if they are law, because they're guidelines and they're open to interpretation, and some of them are very philosophical. I said more than 2 lines, but
no, I think potentially though, you sit on different sides of the table, Robert and Lee. There's an alignment in there in that if the model isn't appropriate or available, it's not necessary, and that you should be able to justify the assays that you're putting forward to the regulators. So I definitely think there's alignment in what you're both saying there. Okay, well we've kind of run out of time for the panel discussion there. Oh no, we have had a question. We've had a question from the audience, so let's just address that last one there. So it's from Ying Dong. Thank you for your question. So this person says, how do you de-risk gene editing therapies in preclinical studies?
I think it's a tough one to answer in a couple of seconds. I think, you know, I'd want to know what genes they were editing, whether they were replacing with a functional gene, if they're gene editing cells to remove, you know, different HLAs and things. you need to understand that therapeutic, and then you'll look at what you're editing, and then you'll say, okay, so what potential issues could they cause clinically? And then you kind of work back and say, is there an experiment that we can do to mitigate this non-clinically, or is it part of our clinical trial management plan? Obviously, gene editing is on the higher end of risks because there's a lot less of it done to date. But as we see more CRISPR products and things coming through, I suspect we'll get a little more comfortable around them as we've got clinical experience.
I think I'd say that it is, as Lee's saying, it's going to be it's not a question that you can sort of answer in generic terms. It's very focused on your specific product indication, target, and everything else. Gene editing itself has been around for decades. CRISPR is just the latest cool incarnation of it, but people have been doing gene editing with zinc fingers, talons, and things like that, and have been in the clinic with those products like Sangamo and others. So there's a whole heap of things that you'd need to get into there, but it will be very specific to your product, your target, your indication, and as Lee was saying, that risk-based approach.
On the subject of gene editing technology, I'll just chip in to say, as Mustafa says, CRISPR is the cool new technique, and obviously we saw the Nobel Prize being awarded to the discoverers recently. It's also a cautionary tale around IP in that the initial early work that was done there was done with, I think, 2 main groups, and as such there are now 2 major competing claims to the fundamental IP that governs that platform. And it's an interesting case because the framework of the IP law is slightly different in the US than it is in most of the rest of the world, and so rights are available in some jurisdictions from one party but not in others from the same party, so that can potentially cause a very complex landscape for somebody who wants to come along and say work with license rights from those rights holders. Even identifying who is the correct person or company to open negotiations with is potentially complicated. And I think one of the main points I wanted to emphasize in my talk as well was these questions are potentially incredibly complicated, and it's really important to engage with your tech transfer office if you're based at a university, or to take that forward with your patent attorney if you're in your spin out phase or your SME phase, and get the professional advice to identify what those issues are, because it could be very costly to not understand where you lie with respect to third-party IP in those decisions.
Thanks, Matt, for that, and the other speakers. I'd just like to say, Yin and anyone else who has any questions, we can by all means set up some one-to-one meetings with some of the experts from the Catapult if you want to kind of discuss these issues further. That's not a problem. We can do that. But I'd like to wrap up the roundtable discussion there, and I just want to share my screen finally, just to say how you can collaborate with the Catapult further. So let me just bring this up. So I internally lead on the what's called commercialization of research, which is an internal funding scheme we have at the Cell and Gene Therapy Catapult. And the idea is to provide in-kind resource to academics to basically accelerate the commercialization of their technologies. And so what we're trying to do, what we describe it as doing, is developing investable propositions and then taking those propositions with the academics to VCs. So this is in-kind support. And we're not asking for anything in return for this. It's just a service we provide. And the remit we've been tasked for by Innovate UK to kind of support the academic community in accelerating the commercialization of ATMPs. So if you'd like to kind of speak to me or anyone in the Catapult about that, I'd be more than happy to have a conversation about, and we can discuss your particular issues and how we can support your technology. And then a final word just to say thank you very much. Thank you very much for joining. This whole presentation, including all the rooms, were recorded, and so what we'll do is we'll share that on a YouTube link in the next few days, so you can go back and look at any of the speakers or any of the presentations at your own leisure and at your own time. So I'd like to also say thank you to all the speakers. Your input was very valuable, and I hope everyone learned something today and took something interesting home. So thank you very much and goodbye.